So like the clip that we just watched, this lesson is about DNA replication. And up until today, I just told you when it happened, not really anything about how it happened. So that's the point of this lesson, is how it happens. The first little bit is about what she said right at the end of the clip. DNA replication is called semi-conservative. Anytime we have a new molecule of DNA, one strand, so one side, will be the original molecule, and the other strand, the other side, will be the new one that that DNA polymerase enzyme built. So it's called semi-conservative because each new molecule conserves or keeps or hangs on to half, semi, of the original. And this happens during, like we already knew, the S phase of interphase. So I have a little picture on the side, and it has actually quite a bit of detail in it. It's the same legend as the pictures from yesterday. You can see there's little pentagon shapes for the sugars, little circles for the phosphates. But what I really want to highlight here is this idea of old and new strand. If I consider my original DNA molecule, I'm talking about sort of this piece at the top here where the two strands are still attached to each other. You can see that the bases are paired up with their complementary base. Then, kind of halfway down, it looks like it's splitting open, just like she showed in the video. We separate the two strands. If you follow this, this piece here is part of the original DNA. So if you see what's happening, I took the two strands and pulled them apart. And then we start adding these new pieces on the inside. So when I'm finished with this process, like if this entire strand of DNA gets replicated, what it would look like if I were to look at the two strands is that the outside of each of them would be the old original strand, and the inside of each of them would be the new piece that was added on during DNA replication. So the point of this picture really isn't actually the detail of the nucleotides, but rather where the old and the new strands are. Now we're going to go through how this all happens by naming all of the enzymes and everything, but does anyone want to ask something about this, the idea of it being semi-conservative? So this little picture is the same one, like it's on the next couple of slides. So I'm going to use the same picture for all of the rest of the information. The picture has some numbers on it, and I will go through and name what all of the numbers represent. But before we even do that, there are a couple of terms that didn't have a number on the picture that you need to know. The first is origin of replication. If DNA is going to make a copy of itself, we need to know where to start. Those enzymes need some sort of signal that this is where replication needs to begin. And the way they get that signal is by looking for a specific nucleotide sequence. So a specific order of A and T and C and G will tell those enzymes this is the spot where you start. Now it came up as a little bubble in the video. She didn't actually really talk about it a ton, but there was mention of how prokaryotic cells and eukaryotic cells are a little bit different. Things like a bacteria, things that don't have a nucleus necessarily, but that still have DNA, might have one small circular chromosome. In a small circular chromosome, like what a bacteria has, there's probably one origin of replication, one spot on the circle where the enzymes get the signal to start replicating. If we take our chromosomes, linear eukaryotic chromosomes, there are thousands on the same chromosome. So when I show you this picture of replication happening, I'm showing one spot on a chromosome. And if we were to zoom out, it would be happening concurrently at like 1,000 different spots along a chromosome all at the same time. That's how DNA replication can happen so quickly. I know it still takes a few hours for it to happen, but that's actually pretty quick in terms of cells getting things done. The other thing that doesn't have a specific number, but that is what kind of the whole picture is about, is the replication bubble. The replication bubble is what this picture is. It's the point where you can see the separation of the two strands of DNA. So on this picture, these dark blue lines, those are DNA strands. This one omits the details of nucleotides, like it's not showing the sugar and the phosphate and the base separately, so that we can focus on some of the other details. So wherever you see this sort of oval bubble-like shape, that's the replication bubble. On each end of the bubble, there's a fork, a replication fork. This picture shows one replication fork really well. Right about here, where the bubble starts and the road kind of forks, maybe think of a tuning fork in music. I think they have two prongs on them. One goes one way, one goes the other way. That's the replication fork. The replication bubble really is where there are two strands of DNA that instead of being attached are unwound, separated from each other into these single strands. And those single strands are used as models for us to manufacture new strands. Okay, so this is the same picture, but now we're going to start labeling things on the picture. First thing for the helicases. Now remember, I'm only showing you one in this picture, but it happens at multiple spots at the same time along a chromosome. Like she mentioned in the video, ends with ASE, so you know that sounds like an enzyme name, and that's what they are. On the picture, here's number 1. Now, is helicase actually a big giant oval? No, this is just a nice way to represent what it looks like. On the video, she showed it being like a zipper. There's no zippers anywhere. There's no zipper prongs. That's just an analogy of what it's doing. These helicases will find the origin, the place where we're supposed to start, bind to the DNA, and then separate and unwind the two strands. If you look, there's an arrow on the picture. That's which way the helicase is moving. And you could tell, even if there was no arrow, that it's moving to the left because the two strands are already separate over on the right. But on the left, the two strands are still together. So that's where helicase still has work to do. Now the second thing on the picture, and you'll see it all over the place, DNA polymerase. Just like helicase, there's multiple of this enzyme, but unlike helicase, in this one snapshot, you can see multiple copies of it. That's because there would be several DNA polymerase enzymes all working at the same time. So it's kind of the blue rounded corner squares that we're looking at for DNA polymerase. If you look at the name, maybe someone remembers what poly means when it's part of a word. Many, lots, a bunch, more than a few. DNA polymerase is an enzyme that's going to add a bunch more DNA nucleotides to our strand. So it is an enzyme. No, it's an enzyme because I looked at its name and I thought, oh, it ends with ASE. That sounds like an enzyme. So it goes into the replication bubble where DNA has already been pulled apart and begins to add nucleotides. And the way that it adds them is by making a complementary strand. So she talked in the video about how DNA was its own spell checker. DNA is looking what nucleotide is already there, and then DNA polymerase brings the complementary nucleotide. Now, someone might ask me, where are these nucleotides coming from? You have nucleotides kicking around. They come from the food that you eat, from other cells that you have in your body that have been decomposed. Every cell that's alive has DNA in it, and thus the components that you would need to build or to have nucleotides. And it mentions here, but I will mention it again, there's a particular way that DNA needs to be added. DNA polymerase has to go in a particular order, particular direction. I don't know if any of you shovel the snow on your driveway. I have to because I'm a grown-up. I have to shovel my own snow. It sucks. But I am right-handed, so when I hold the shovel, I hold like the top of the shovel in my right hand and then the bottom of the shovel in my left hand, because my left hand's not useful for like controlled movement, it's just there for a lever. So when I'm shoveling snow, it makes way more sense for me to go shovel off this way and then take a step back and then shovel again. It would not make any sense for me to do it in the other direction because I'd be throwing snow on the place that I already shoveled, which is silly. So I'm giving you that example because that's kind of how DNA polymerase is. There's a way that it makes sense, just the way things are oriented, to continue to add new nucleotides. It's always going to start at the 3' end of the original strand. So if I look at the DNA polymerase up here, these two squares, it's showing it over here now, but it started over here. That's where it started making that new, slightly lighter blue strand on the inside. So DNA polymerase has to go in a particular direction. The next two pieces kind of go together. We have primase and then the primer. The primase is number 3 and the primer itself is number 4. And you can see down here there are more of them. Now this picture is only showing the primer being made on one side of the DNA molecule, even though in the video she showed it on both sides. And that's because this is where it's a bigger deal. So that's why my picture is showing it here. Primase ends with ASC. So it's an enzyme, just like DNA polymerase and helicase. And I need this primase enzyme to make a primer. So I'm going to skip to what the primer is and then come back. What's the primer? It's a short strand of RNA that gives us a starting point. DNA polymerase needs to know where to begin. Where should I start putting new nucleotides? And the primer is that for DNA polymerase. If you think about primer in other sense of the word, maybe some of you have painted something before, like a wall in your house, and you just put primer on the wall before you put the paint on. Sometimes there's primer mixed in with the paint, but it helps the paint stick and stay on the wall so you get nice, clean-looking paint coverage. So the primer is going to help the DNA polymerase stick, give us a place to put our new nucleotides. So primase is the enzyme that's going to attach this little segment of RNA just so there's a starting point, somewhere to begin. Eventually, DNA polymerase removes the RNA as it starts adding new DNA nucleotides. So we have 3 enzymes: helicase, DNA polymerase, primase. She mentioned some other stuff in the video, like single-stranded binding proteins and topoisomerase. That's beyond what you are required to know. But these are the basics. These are ones that realistically you could be asked about, like a test question, on a diploma question. These are the must-knows. Now there are a few more things we need to label, but before we do, does anyone want to ask about any of those 4? What they do, where they are in the picture, anything like that? So then last little bit of stuff is what 5, 6, 7, and 8 are. You'll notice that 5 and 6, I just named them because my description of the other 2 tells you what they are. Number 5 on the diagram is ligase, and you see it down here. It looks like a little yellow circle. On the video, she called it the gluing enzyme. And at one point, it's ligase, and it says, glue all the things. And I can just imagine a cute cartoon voice, like kind of alien sounding, saying, glue all the things. Then number 6, you can see there's number 6 here. It's pointing at this arrow. I could have put a number 6 on any of these little fragments of blue on the inside. Those are Okazaki fragments. And then we have number 7 and number 8. Number 7, right here, is this whole strand of DNA being made on the inside. It's called the leading strand. And if you look at what's happening, it is moving in the same direction. It's being made or synthesized in the same direction that helicase is moving. So we're leading, we're going in the same direction. You'll also notice that it's continuous. So there are no breaks in that strand, there are no fragments. That side of the DNA molecule is oriented so that DNA polymerase can start on the right and just add new nucleotides because things are in the right direction. New nucleotides must always be added starting at the 5' and ending at the 3' end. So on my original strand, that's the 3' end. That means my new strand is going to be anti-parallel and start at the 5' end, which is what DNA polymerase wants. The other strand, number 8 down here, is the lagging strand. It's actually all of the Okazaki fragments all together. The lagging strand is replicated in short segments. So that's why instead of one continuous line, there are a bunch of little lines. You'll also notice if you look at the arrow that it's pointing the other way. It's not going the same way that helicase is going. It's not going the same way that the leading strand is going. It's going to the right on this picture because DNA polymerase can only work from 5 to 3. It has to go in that direction. If I look at my original and new strands, this is the 5' end of the original strand. That's the 3' end of my new strand. So DNA polymerase has to go from 5' over here towards 3'. It can't go the other way. So like in the video, while DNA polymerase is continuously adding new nucleotides on the top, we're making these little fragments along the bottom. In the video, it showed one enzyme racing to the beginning. That was a simplification. There's actually multiple copies of the enzyme working at the same time, but it's a good way to visualize what's happening. Eventually, those little fragments get linked by our ligase enzyme. If you look at the name for it, maybe some of you, like me, enjoy crime TV shows where detectives are looking for ligature marks. And so if you know what the word ligature means, it's like tying something up. That's what ligase is doing. It's tying up the pieces or the fragments, putting them together.